Hello, everyone, and thank you for joining our session today. My name is Guillaume Béchade, and at Wados, I take care of biopharmaceutical activities in Europe. In the next half hour, I'll be delighted to share with you why we believe the biopharmaceutical analytical community can benefit from more accessible and deployable LC and high-resolution MS solutions for routine characterization and monitoring. The biopharmaceutical industry is currently living profound dynamics. Biosimilar, for example, are spurring intense competition and driving the need for more greater productivity among developers and manufacturers. This in turn is fueling investment in new development and manufacturing technologies, like, for example, greater MS downstream, continuous manufacturing, etc. It also pushes towards modern analytical approaches like quality by design or the well-known multiple attribute monitoring that can provide a competitive edge. Secondly, outsourcing the development and manufacturing of biologics continues to expand. Actually, outsourcing partners work on a cost plus service model. They are not earning revenue from the sale of the drugs being manufacturing, but on a markup of their services and the services they can provide to their partners. Hence, to compete, they need to be as productive and as cost-effective as they can be. The complexity of biologics is only increasing, and as more and more products come to the market, there is a need for better analytical tools that can confirm the biophysical characteristics and the identity of these highly complex molecules. All of this with greater accuracy and greater precision. And finally, efforts are also being made to harmonize regulatory standards globally. Import-export rules, for example, are in many cases favoring local production, so manufacturing and quality release testing needs to be replicated in multiple geographies. In terms of technology supporting these trends now, MS in the biologics license applications field has been increasing in the past 2 decades. For example, from 2000 to 2015, almost every BLA had mass spectrometry data in filling. In addition, the number of attributes measured by DMS over the time has been steadily increasing. FDA itself recognized that additional MS methodology is seen within the quality control and comparability section of the filing. It also appears that there is an evolution of the LC-MS usage from non-regulated environment to the highly regulated and compliant environment. Within biopharma companies, MS technology, as you see on this chart, is generally accessible through expert central or support labs, owning the full characterization as well as the routine monitoring work. Many samples and lots of information flow between these busy labs and the other teams taking care of bioprocess development, stability, or QC testing, for example. This situation creates major bottlenecks as business pushes to shorten time to biological license applications field. The central labs have also some difficulty to put in place data integrity or GMP compliance. They have also limited capacity, and this is a strong constraint, requiring sometimes to outsource as the workload cannot be coped with. Therefore, support labs are generally waiting for results. That creates a strong bottleneck, and also the move of data across labs and geographies can present a certain difficulty. So when we ask the community what would allow deploying MS beyond core labs, the top 5 requests result in a product thinking that is radically innovative in terms of a system. Foremost, the system needs to be robust and reproducible so that consistent results can be generated rapidly. Time to learn a new system and time to get to results needs to be minimized. Also, laboratory space is an operational cost in the company that should be reduced definitely, and modification to facilities should be minimal. Lastly, reducing the complexity and time to get actionable informatics is highly desired. Within that backdrop regarding the biopharma industry and its increasing need for MS analysis, that can be made across the drug discovery and development pipeline, we at Waters have been very pleased to introduce the BioAccord system, the first smart MS-enabled biopharma LC-MS solution. It's a purposefully designed small benchtop system that incorporates smart MS technology for maximum ease of use, deployability, and accessibility. As a time-of-flight instrument, it provides accurate mass data, and it is optimized for routine biopharmaceutical analysis, including peptide mapping, intact mass analysis of proteins or oligonucleotides, and release glycan analysis. As a workflow-driven system, it enables automated data acquisition, processing, and reporting, so you can get results more quickly and make faster decisions. It is also compliant-ready, so you can be confident in meeting regulatory requirements. So what is exactly this smart MS concept and why it is important? Well, perhaps unsurprisingly, it turns out that with analytical instrumentation, and as in many other areas of our lives, the users are looking for tools that are faster, that are simpler, and that are more reliable, and at the same time still being able to perform the job that is required. The view is that specialist training and years of experience should not be needed for any but the most advanced equipment. Modern systems should provide an intuitive user experience, and any complex tasks should be automated where possible. That's how we came to develop the new system with these smart features designed to help any user with the minimum amount of training to get the system up and running, understand what's happening, and even be able to recognize and resolve issues by themselves. So let's have a closer look at some of the functionalities of this smart MS. You see here the setup window of the instrument. It's been simplified to the extreme, with 3 buttons to start, stop, or reset the setup control, and only 4 options are available to set up the system: positive or negative ionization. Positive, stop, and start. So you see here the setup window of the instrument. We have simplified it to the maximum, with 3 buttons only to start, stop, or reset the setup, and 4 calibration options to simplify the workflows at the maximum. You have positive or negative ionization options, and low mass for peptide mapping or oligos up to 2000 mOV, or high mass up to 7000 mOV for intact protein or oligo analysis. The system is equipped with interactive displays that can reassure the user on the status of the equipment and also guide him or her in terms of any diagnostic checks that needs to be performed. In case of an issue with the system, the display on the front panel will display a check icon with a pictogram that indicates the type of issue. For example, here, a leak has been detected. On the PC screen that is associated to the system, the easy resolution guidelines will be also displayed in very simple steps, so the user can be guided through the resolution of the issue. Then the system will retest if the issue has been solved and give a green light to the user, or show a second level of issue resolution. The software is completely a part of the solution. You see here that the software we use is called Unify. The BioAccord system is directly controlled by this platform that integrates the sample information, the creation of method and sample sequences, the control of the system, as I just said, the processing of the data, and finally, the reporting and data management capability. This unified software is based on a Oracle database that will enable all the 21 CFR Part 11 compliance requirements. As you see here, the full audit trail, multiple systems can be installed on a network architecture, so secured data can be ensured. Compound libraries are available and can be customized. User roles and rights management is completely integrated in the platform. The system can be qualified for GXP labs. Central repository of workflows and standard operating procedures can also be implemented in the platform. And finally, the reports can be electronically signed at multiple levels, ensuring the perfect traceability of the information in the platform. Now, in terms of application, as I said earlier, the system is designed across 4 different workflows: intact and subunit analysis, peptide mapping or multiple attribute methodology, and release glycan. The intact mass can be operated in denaturating or native mode. Denaturating will give you access to mass confirmation for your proteins. This analysis will also give you proteoform heterogeneity profiles, while the native protein analysis, for example, using ion exchange chromatography, can give you access to charge variance measurements. We also make available size exclusion chromatography coupled to MS, so for the native analysis. Subunit analysis, after reduction of the monoclonal antibody or digestion using IDS type of enzyme, can go a little bit deeper in the primary structure and the post-transactional modification verification. And if you operate that in the native, you also have access here to more precise information around the acidic or basic impurities. Peptide mapping or monitoring will give you access to the primary structure and the modification verification and relative quantification. The site-specific profiling and monitoring of post-transactional modification is also made accessible through this workflow. Next, release glycan analysis will give you access to glycan structure, the profiling of this, and the relative quantification of the different glycans on your protein. And finally, I mentioned oligos as a workflow that can also be supported by this platform. This would, for example, give access to mass confirmation of any oligo that are produced. And we are developing capabilities in that area in the next future. Let's have a deeper look at the intact and subunit mass analysis workflow. You see here that we really cover each and any step of that workflow, from specific installation kit for intact and subunit mass analysis, that making use of a monoclonal mass check standard that is provided. We offer a selection of consumables, separation columns like the Acuity UPLC protein VEHC4 column or the BioResolve reverse phase monoclonal antibody polyphenol column. Native MS is also enabled through the EH size exclusion chromatography columns or the BioResolve SEX map column for ion exchange and charge variant analysis. The system is equipped with a UV detector and with the time of flight mass spectrometer. And now the results. You see here on the left-hand side the raw spectrum that we could obtain with the platform for the monoclonal antibody mass check standard that we typically use to set up the platform. You see on the right-hand side the deconvolved spectrum that give access to the different proteoforms that the monoclonal antibody can present. And the system will process this information and extract the different glycan identities that are present in the sample, showing also the percentage for the different glycoforms that are present in the sample. So you see on the right-hand side of the table on the screen, the direct measurement of the relative glycoforms quantification that is readily accessible. So you get both mass confirmation for your sample, glycan identification and quantification. One of the key requirements from the users was that the system could be reproducible and repeatable. You see here an extensive reproducibility study that we have performed on the system over 5.5 days. We have injected more than 1,000 times the same monoclonal antibody standard to check the reproducibility in terms of relative glycoform quantification. You see on the trend plots here that the measurement is very, very stable, and the RSD values are astonishing. Less than 2% variation across these 5.5 days for such precise measurement. So you get a consistent relative quantification of these observed glycoforms. So you could say it's true on one system for one sample, but what about multiple units actually? Well, we've challenged that, and we have made the same analysis over 6 different BioAccord systems with 10 injections per system. And you see that we also get here very, very good reproducibility, showing that the relative quantification of the glycoforms is consistent across the different units of BioAccord. When the data has been processed and reviewed by the user using very streamlined workflows that can be customized, you can also generate reports that can, of course, be also fully customized. Here is an example of the combination of chromatographic data, raw NS spectra, process NS spectra, tables data, as well as trend plots. All of this can be designed as a template and made readily accessible for the routine users. So that was for intact mass and subunit mass analysis. Let's have a deeper look now at peptide mapping. We also cover the full workflow with a specific kit and specific standard using a monoclonal antibody triptych digest that is used for checking the performance of the system, and that can be a good system suitability testing sample as well. There are choice of consumables in terms of UPLC peptide columns that can be chosen from. The system used is the same, and you still use Unify for the acquisition, processing, and reporting of this data. So you get a little bit more complex chromatogram across this 1 hour separation here. You see all the peptides very well separated using the UPLC system. All this information is processed automatically, so the peaks are integrated, the data is extracted, and the coverage sequence of the monoclonal antibody is automatically calculated, as you see on the right-hand side. In this sample here, we get 96% coverage with quite strict criteria in terms of mass tolerance, number of fragment ions that can be identified, and the minimum length of the peptides considered were 4 amino acids. I mentioned fragmentation ion because this system, although it's a tough system only, has capability to perform data-independent analysis fragmentation experiments that give access to the amino acid sequence and the localization of the post-transactional modifications. All of this is processed automatically by the software. As you see here, all the peptides are listed in a table with the possible modifications that they exhibit, and the percentage of modification is automatically calculated through the system. For example, on line 60 and 61 here, you see that the peptide 42 is presenting a leading truncation that is evaluated around 83% of the peptide 42. Extracted ion chromatograms can be generated for these 2 peptides, and fragmentation information is automatically displayed and, of course, processed. So you get confirmation of the amino acid sequence and possibly the position of the modification. Another example is the study of an oxidized peptide. For example, here, the peptide 21 in the sequence is containing a methionine that can be oxidized. The system will evaluate the level of this oxidation compared to the native peptide, confirm the position of the modification using the fragmentation data, and possibly look at the trending of the of the variation of these oxidations through a batch of samples. You see here 25 replicated samples that show only a variation on this oxidation level of less than 2%. So very good data reproducibility here again. This test on one peptide, one attribute, or multiple of these attributes can then fully be made automatic. So the percentage of the modification can be monitored across various samples or assays. You can set up limit check levels. For example, here, the peptide that is oxidized was required to be less than 50% oxidized, otherwise an error would be displayed. For example, the red bars over here. Or if it's below 50% but higher than 10%, the system can also give a warning to the operator. I'd like to turn now to the release and glycan analysis workflow. You see here that we can provide also the full workflow from sample preparation using the RapidFluor MS and glycan kit, the separation consumables using a UPLC glycan VEH amide column operating in the HELIC hydrophilic chromatography mode. And the system in that case can be equipped with a fluorescence detector for quantitative analysis, and the mass system plus the informatics tools remaining the same. Traditionally, using 2AB or 2AA labels for glycan analysis is a very tedious and work-intensive process. It typically takes from 5 hours to 2 days to prepare the sample to be ready for release glycan analysis. We have challenged these situations, and a couple of years ago, we have introduced the GlycoWorks RapidFluor MS and glycan kit as a way to have a very fast sample preparation process. Deglycosylation can be as fast as 10 minutes, and it's total. The labeling with our RapidFluor MS label takes less than 5 minutes, and the purification on the GlycoWorks HELIC macroelution plate is also can be done in 10 minutes. So overall, within 30 minutes to 1 hour, if you account for the pipetting steps, your sample is ready for injection on the LC-MS system. The result of this is very sensitive fluorescence detection, as you see here, and also very sensitive MS response, thanks to the design of the RapidFluor MS label. You see here that the software has identified all the peaks corresponding to different glycans, and the relative quantification of these glycans is made possible through the fluorescence signal. So this workflow is enabling the user with a very fast sample prep, less than 1 hour, very good sensitivity both in fluorescence and MS, as you see here compared to the traditional 2AB or procainamide, as well as instant AB. The sensitivity is much higher in both fluorescence and MS. Chromatography keeps the very good quality in terms of reproducibility, even if it's the more challenging HELIC separation modes here. We get a reliable identification using glycan library that are provided with the Unify software, and that making use of the MS data to confirm the assignment of these glycan species. And finally, the data analysis is made quick thanks to the Unify workflow and the very customizable reporting capability. Yet sample prep is still a manual process in that case, and many users are considering automation capabilities as a way to gain into reproducibility and efficiency in the lab. So we have enabled the RapidFluor MS glycan kit to be compatible with automated sample prep platforms like Hamilton or Tecan, or Andrew Alliance that has recently been acquired by Waters. So we provide for these platforms verified automation scripts and spec layouts, purposeful kits for automated liquids handling with the appropriate volumes, and scalable sample processing from 8 to 96 samples at a time. The Andrew Alliance platform, called Andrew Plus, is a very flexible pipetting robot that ensures reproducibility, traceability of the results, and as well maximize lab efficiencies and resources. It's a very easy to use platform that allow you to rapidly create protocols without any programming knowledge or dependency on specialist staff. Anyone can really use and program that robot. It's a versatile platform that scale or adapt the workflows by simply swapping Domino consumables orders on the table. That can also be considered for connected tools or connected devices through one lab, a very easy to use software. It's also very easy to deploy thanks to a method transfer by cloud native software that will ensure the maximum productivity with minimum downtime. And finally, the application knowledge is growing and growing around that platform. So you benefit from constantly expanding libraries of protocols that are available online, and you can get up to speed very quickly with it. Of course, the results need to be consistent across various methodologies. We have compared here the different glycan capabilities, glycan prep capabilities. So we observe consistency in the results. You see in blue the manual sample preparation process. In orange, Series 2, the Andrew Alliance robot. And in green and yellow, the Hamilton and Tecan robotics platform that have been used to prepare the samples. And you see the results for different glycans are extremely consistent across these 4 different ways to prepare the samples. So that brings me to a conclusion. Our vision for the biopharma industry is really to increase the lab's throughput by segmenting the full characterization from the routine monitoring. The full characterization can still be operating in central analytical labs where routine assays could be performed on a platform like BioAccord and transferred to bioprocess teams, analytical developments, stability or QC labs. So the efficiency is dramatically increased in the overall organization. This dramatically reduces the wait time for bioprocess labs to get results. It also reduces the risk by being able to run more, if not all, desired samples at the different stages of development of product or production. And finally, the development and the transferability of methods is made easy also for non-expert users that are able to run successfully these methods very close to the decision point in the process. So here we are, the BioAccord system. That's actually a multi-award winning solution. It gets the Frost and Sullivan Biopharmaceutical Analytical Instrumentation for the New Product Innovation Award this year, as well as the well-known Select Science Best New Drug Discovery and Development Product of 2019. We have very good customer feedback on this platform. I encourage you to have a look at Select Science website or on our website, www.waters.com/bioaccord. And you can also have access on the Select Science platform to this application notebook here, telling a little bit more about mass detection and how this can enable faster biopharma analytics. Thank you very much for your attention this day, and we'll be very happy to answer any question and answer in our virtual booth online. Thanks for watching. Bye-bye.